So tell me honestly, have you ever walked out of physiology exam thinking, I literally studied everything, then why my marks are average? Because you are doing the same mistake that most of the average students are doing, writing answer in this format. They are too long, where are the headings, no flow, and land up in getting 4 out of 10 marks. Now look at this masterpiece by a topper. The same answer, all few clean, neat pages, and all points under the headings and excellent structure. What is the difference? It's not IQ, not luck, it's presentation. And in this video, I will show you exactly how toppers write Neural Regulation of Respiration in university exam, step by step, by using flowchart, diagram, clear headings, and examiner psychology. Now look at this question carefully. Name the respiratory center, explain the neural regulation of respiration, and add a note on Hering-Breuer reflex. The biggest mistake that most of the students do is, the moment they see long essay question, they become panic. But the toppers do something different. They immediately divide the answer into headings like respiratory center, neural regulation of respiration, and note on Hering-Breuer reflex. The moment you divide the answer, your brain becomes calmer, and the examiner understands that your answer is organized. For all long essay question, follow this 10-mark blueprint. Always maintain this sequence. First, definition and introduction. For this, keep it in brief, maximum 3 lines you can write. Next, classification and broad flow chart. Show the big picture in the beginning of the answer only. And main explanation with subheadings. Use bullet points. Next, anatomical and conceptual diagram. Draw it with a pencil and label always on the right side. Next, comparative tables if applicable. And lastly, the applied or clinical physiology. Remember, this is very crucial for the distinction. It's very important point to write this blueprint. If you master this, your half work is done. Never start your answer directly. For best presentation, always start with the introduction. In this, respiration is an involuntary function that can be influenced voluntarily. So thus we are having two control. One is involuntary control and second one is voluntary control. Involuntary control is mainly located in brainstem. Their function is mainly genesis of spontaneous rhythm, whereas voluntary control is located in cerebral cortex and it helps in talking, singing, swimming and breath holding. So ultimate goal of regulation is adjust total ventilation to match the metabolic needs. See, introduction should be small and it should include maximum 2 to 3 lines. Now here comes the main part of the answer. That is, name the respiratory center. This is where the toppers score more marks than the other students by writing this in a flowchart and not in a paragraph style. So respiratory center we're having 2. One is medullary center and another one is pontine center. There are 2 medullary center. First is dorsal respiratory group and second ventral respiratory group. Pontine again 2, apneustic center and pneumotaxic center. Now dorsal respiratory group having 3 neurons, inspiratory neurons, inspiratory off switch neurons and integrator neurons, whereas ventral respiratory group we're having 2, inspiratory neurons and expiratory neurons. So in less than 30 seconds, examiner understood your structure, and remember, structured answer always look intelligent. Now after flowchart, this is the next area to score maximum marks, that is a medullary and pontine respiratory center. This diagram shows dorsal respiratory group and ventral respiratory group of neurons in medulla, and apneustic center and pneumotaxic center in pons. Remember, while drawing this diagram, always use pencil, never pen. Keep all the labels strictly on the right side. Draw a box around the diagram and always write caption figure below it. Now here comes the third part of the answer, that is medullary respiratory center. It generates the basic respiratory rhythm. It consists of dorsal respiratory group of neurons located in the nucleus of tractus solitarius, and it contains mainly eye neurons. It functions as an upper motor neuron for respiratory muscles. That is, it crosses the midline and supplies mainly 2 muscles, that is diaphragm and external intercostal muscles. The inspiratory ramp signal. This signal is not a sudden gasp, but steadily increases for 2 seconds like a ramp and causes steady increase in lung volumes and abruptly ceases for 3 seconds, causing the expiration. You can see from the diagram, dorsal respiratory group of neurons supplying mainly chief muscle of inspiration, that is a diaphragm, and this showing the ramp signal for 2 seconds and abruptly ceases for 3 seconds, causing the expiration. Now let's see how does this ramp signal end. It works via 2 types of neurons, that is inspiratory off switch neurons and integrator neurons. In this inspiratory off switch neurons, they terminate the activity of eye neurons. Thus, it turns off the excitation to the inspiratory muscles, then the muscle relax and causing elastic recoil of lungs and the expiration begins. Whereas integrated neurons, they are mainly stimulated by the eye neurons, and when depolarized to critical level, they fire the inspiratory off switch neurons. Thus, integrated neurons, they receive the excitatory inputs from cortex, pneumotaxic center and from the vagal efferents, whereas they also receive inhibitory inputs from mainly the apneustic center. This is how the dorsal respiratory group of neuron work. Remember, DRG neurons play active role in quiet involuntary breathing. Now the ventral respiratory group. These contains mainly both types of neurons, that is inspiratory neurons and expiratory neurons, and it supplies the internal intercostal muscles and abdominal muscles, that is the muscles of expiration. What is their role? They are totally inactive during quiet or normal breathing, but they become active only during forceful respiration, that is during heavy exercise. What is the interaction? They show a reciprocal innervation, that is I neurons and E neurons both have mutual inhibitory connections. If I neurons are active, the E neurons are inhibited and vice versa. You can show their interaction with the help of this diagram. After medulla, we move upwards to the pons, that is a pontine respiratory centers. Here we are having two respiratory centers, one is apneustic center and pneumotaxic center. This is the part where most of the students confuse themselves, but I will clear it in 10 seconds. Apneustic center. Remember, apneus for accelerator. It acts like an accelerator, whereas pneumotaxic center acts like a brake. Since apneustic center acts like an accelerator, they send the excitatory signals to the integrated neurons, and it prevents the switch off of inspiratory ramp signal. So it produces the apneusis. That is, it causes prolonged inspiration. Whereas pneumotaxic center, it acts like a brake. They are located in upper pons, and main function is it inhibits the apneustic center. So it shortens the inspiration that lead to the shallow, rapid breathing pattern. You can see from the diagram, in upper pons we are having pneumotaxic center, and in lower pons we are having apneustic center. Brakes above the accelerator. And lastly, the reticular activating system. It stimulates the centers to increase drive, which diminishes during sleep. You can use these reciprocal arrows to show the physiological relationship instantly. That is, pneumotaxic center, it acts like a brake, so it inhibits the apneustic center. And as I said, apneustic center acts like an accelerator, so it stimulates the inspiration, causing the prolonged inspiration, that is apneusis. Now, when you are writing the third part of the answer, this is the most common mistake that all of the students do. Look at this answer carefully. Where is the margin? There is irrelevant history. The answer is too crowded, and the examiner cannot read it properly, and ultimately it ends up in giving only 2 marks. Instead, just focus on the physiology. Write all these headings under a bullet points and improve your presentation. Now, Hering-Breuer reflex. This reflex is because of the efferent from the lungs. The receptor for this reflex are pulmonary stretch receptors, which are located in the smooth muscles of bronchi and bronchioles. And these receptors are mainly stimulated by the inflation of the lungs. Whenever lung inflation increases, it initiates the Hering-Breuer reflex, which inhibits the inspiration to prevent the overinflation of the lungs. Remember, this reflex is weak in adults. That is, it operates only when if the tidal volume is more than 1.5 liters. Now, along with these pulmonary stretch receptors, we are also having one more receptor, and that is called as J receptor, also called as juxtapulmonary capillary receptor. You can see the location of this receptor in this diagram. It is in the interstitial space. Now, these J receptors are unmyelinated vagal C fibers near the pulmonary capillaries. This receptor is discovered by an Indian scientist, that is A.S. Pentel. These receptors are stimulated whenever there is increase in the interstitial fluid. In which condition this interstitial fluid increases, like conditions pulmonary edema, pneumonia and congestion. So whenever this interstitial fluid increases, it initiates this J reflex. That is, it causes apnea followed by tachypnea, rapid shallow breathing and bradycardia and muscle weakness. Both discourages the exercise. Remember, these pulmonary stretch receptors and J receptors both play an important role in protective reflex. To conclude the answer, we are going to write about the applied aspect. For applied aspect, we are going to write 2 important points. First one, the effect of brain sectioning, and second one about the Ondine curves. First, effect of brain sectioning. In this, the experiment that proves where the respiratory center lives. What happens if we cut the vagus nerve and take a section in mid-pons? This can be explained with the help of 3 levels, level 1, 2 and 3. Level 1, if we take the section between the medulla and pons, the result is basic rhythm remains intact. The proof is medulla alone, it generates the basic rhythm. Level 2, if we take the section between mid-pontine, that is between the pneumotaxic and apneustic center along with the vagotomy, the result is it causes prolonged inspiration, that is apneustic breathing. Why? Because apneustic center stimulate inspiration, but normally it is inhibited by the pneumotaxic center and vagus. But if we cut both, the apneustic center remains unchecked and causes prolonged inspiration. And level 3, section between the rostral to the pons, that is it results in normal rhythm intact. The proof is pneumotaxic center check is sufficient to control the apneustic center. Second part of this is ondine curse. Now this is clinically known as primary alveolar hypoventilation. It occurs when the automatic control centers are disrupted but the voluntary control remains intact. For example, in bulbar poliomyelitis, the result is the patient must consciously remember to breathe, and if they fall asleep, automatic breathing stops, which can be fatal. Now let's review the entire answer quickly. As I said, do not directly start with the answer, start with the introduction. We have seen in introduction we have written only 2 points about the involuntary and voluntary control. Then we have written about the respiratory centers, this flow chart. Along with this flow chart, there is one important point, that is this diagram of medullary and pontine respiratory centers with the labeling on the right side. After this, we have written about the medullary respiratory centers, the dorsal respiratory group of neurons containing inspiratory signals. Now this is followed by the inspiratory off-switch neurons and integrated neurons. Along with these medullary centers, we have written about the ventral respiratory group of neurons containing I neurons and E neurons. With these medullary centers, we have written about the pontine respiratory centers containing apneustic center and pneumotaxic center. And lastly, about the additional note about the Herring-Buehler reflex. And along with that, we also mentioned about the J receptor. Do not forget to write about the applied physiology. In this, we have written 2 points. First one about the effect of sectioning of the brain and ondine curse. Now revise this topic before exam and you are done. Now go and watch this revision video of regulation of respiration to revise this topic in 5 minutes, and another video of flowchart and diagram trick to remember the entire topic before exam. Thank you.